All right, I want to thank the organizers and everyone else who stayed here all the way through the evening. I will also stay in the realm of nuclear physics, although I'm going to go to a much smaller scale than the previous speaker. Just like many of us who were presenting here, we sort of felt the burden of meeting the expectation of presenting in the institute named after Schrödinger, so I struggled quite a bit with the title. What should I name my talk? I don't do quantum mechanics. Of course, doing experiments with vertebrates is not easy. So here's my title: Nucleosome is the hydrogen atom of epigenetics. It's more of a work in progress. So whenever a good student comes along, we make a step and then wait for another good student to come along. There's several students and postdocs that contributed to the story. So, when you look at the nucleosome and you look at the hydrogen atom, you wonder if there is a useful analogy to be made. Of course, if you want to keep up with the times and if you have a lab full of students, you want to do some kind of AI and ask the AI, right? So I did. That's what AI came up with. It certainly is intriguing. I'm not sure if it's useful. I get back to the drawing board, and then one obvious analogy is that both the hydrogen atom and the nucleosome are just fundamental units in their respective fields. All right, what else? What other analogies can you see there? Well, we know that for hydrogen atom, the key to determine its properties are the energy level, electronic energy levels. And then also you know that if you put the hydrogen atom in an electric field or magnetic field, then the energy levels are going to be bent, they're going to be split. This is the Stark effect. So let's see if something like this works for the nucleosome and where this may come from. Now, for the energy levels, the hydrogen atom, the analogy would be free energies of various transitions, structural transitions in the nucleosome. Now, the question is, what can we substitute would be the analogy of a field. Now, I will start with the first question, and then we'll move on to the second one. All right, so for the ionization energy of the nucleosome, well, it's obvious. What does it cost to strip off the DNA? So we looked into this a long time ago, already 13, 14 years ago. Essentially, as was mentioned before, the nucleosome is governed mainly by electrostatics. So you set up Poisson-Boltzmann equation for the system. You do the calculation. The first step we did, just like for the hydrogen atom, the first step was fully analytical. You set up a bunch of cylinders. DNA is a cylinder, nucleosome is a cylinder, the wrapped state is a cylinder. You calculate what does it cost to separate the two, and the ionization energy of the nucleosome came out to be about 60 kT. So what it tells you immediately is the nucleosome is sort of a vault. It explains why the DNA is so well protected in the nucleosome. In fact, it almost doesn't matter whether it's 60 kT or 80 or 100. Essentially, the chances of spontaneous unwrapping of the DNA on its own are pretty much nil. So one other interesting fact that came out of that study is that we looked more closely into the nucleosome. In fact, we calculated phase diagram in the coordinates of the core charge, so histone core charge versus salt, outside salt. And immediately we can see that the nucleosome in vivo sits at the phase boundary that separates the wrapped and the unwrapped state. And in fact, it's only three charges that separate, three unit charges that separate this wrapped from non-wrapped state, which immediately suggests that if you want to fine-tune and regulate the nucleosome stability, this is the charge, the core, the charge of the globular core is the kind of the crank, is the knob that you want to tweak, not the salt, but this one. So what is that? How can you regulate this charge? Well, we all know there are multiple post-translational modifications that can be done that the cell utilizes in the nucleosome. Citrullination was mentioned before. Of course, there is acetylation. There are many more. So acetylation, right? So you have the lysine and it gets neutralized. All right, so the next question we asked, what are so there's this energy splittings, right? So if you were to calculate delta delta G due to various post-translational charge altering post-translational modifications to the histone core, you can do that. You do let's say, acetylation, and you just calculate the change in delta G. We've done that. We've done this using full atomic detail of the structure. This is no longer a cylinder model. So this is an example of a couple of PTM sites. Of course, the nucleosome is symmetric, so each one comes in double. Now, the nucleosome, as we know, it's not a single structural state. In fact, it exists as an ensemble of states. So you have to put them in the model. Again, I'm skipping the details. So we've calculated, the first step, we've calculated those delta delta G is due to acetylation. There's precious little experimental data, but when we put the experiment and prediction on the table, it was reassuring that we were possibly on the right track. So there's this 3 known acetylations, there's experimental delta delta G's, and these are our calculations. The ordering is right, and in fact, we're almost, for these two, we're even within the error bound experiment, and this one we're pretty close. So this is a complex system, lots is going on, so we were pleasantly surprised to see that the calculation yielded something as meaningful as that. All right, so now let's look at, of course, experimentally, it's a really difficult experiment to do, as well as in vivo, just in vivo experiments, in vivo acetylations on the nucleosome now. But in Virtuo, we can calculate them all. In fact, we can calculate the effect of all post-translational modifications, charge-altering modifications, regardless of what they are. So let's look at acetylations. Let's put them all together on the nucleosome on the map. Now, essentially what this shows you is that the brighter the color, the stronger the effect. Now, you can see some of the trends are fairly obvious. So if you acetylate, if you kill the charge, alter the charge of the core near the DNA, of course, the effect is going to be stronger. The destabilizing effect is going to be stronger. If it's closer to the center, the effect is weaker. But some of these, for example, this ones, right, they're pretty close to the DNA, but the effect is weak. Some of them are fairly far removed from the DNA, but the effect is stronger. This is not obvious at all. It certainly doesn't come from a simple cylinder picture. The physics, of course, is the same. It's the physics electrostatics, but it's more complex. It's modulated by the complex structure of the nucleosome and the multitude of states that is there. All right. So from this, we can already see that the first step of our aim of making an analogy with the hydrogen atoms. This is the spectrum, in the sense that the energy diagram of the nucleosome. This is the ionization energy, and these are the charge-altering PTMs. There's a whole lot of them with a different amount of destabilization. Some of them destabilize the nucleosome fairly significantly, and you might imagine that this may be utilized by the cell if the DNA accessibility is to be fine-tuned. But some of them are very light, have almost zero effect, and we speculate that those might be used as passive anchors, where you just need to acetylate something. This acetylation is recognized by something else, but you don't want to disrupt the nucleosome itself. All right, so now, let's try to make connection to function. Of course, this is tentative, and it's very simplistic. The simple connection is essentially if the DNA is tightly wrapped, it's inaccessible or poorly accessible. So whatever depends on the DNA accessibility, such as transcription, is going to be downregulated. When you make DNA more accessible, these processes are going to be upregulated. It's a very simplistic picture, but let's take that and look at the effect of the acetylations. So what you see here is the change in the DNA accessibility versus the acetylation coordinate. In other words, these are all possible acetylations in the histone core. Now, As you can imagine, it's not a surprise that most of them, almost pretty much all of them, destabilize the nucleosome and make DNA more accessible. But as I said before, the degree is very variable. Some of them make a huge effect, some of them have almost no effect. And again, we're making the claim that this is effect on transcription. Now, there is one, however, which is completely counterintuitive. There's one, is this H4K77, I believe. It's predicted to, in fact, make DNA less accessible. So it's completely counterintuitive. You kill a positive charge on the core, so you decrease the electrostatic interaction with the DNA, but then it makes the DNA less accessible. There's an interesting structural story behind it. I'm not going to go into details, but the prediction here is that, of course, this one is going to suppress transcription. So this is a deal breaker for a theory, right? If this doesn't come out right, then the theory is wrong. So we made this prediction. We looked into literature, and lo and behold, fortunately, there's a paper that showed that indeed, for unexplained reason, this one H4K77 indeed results in a repressive phenotype. I forgot which organism was used, but so that came out to be correct. All right. So the next question is, remember on the diagram, I made a claim that we can make a full analogy, not only with the energy levels, but also with the fields. Now, if you put the nucleosome in a very strong electric field, then of course something will happen to it, but the field has to be really, really strong. So in the cell nucleus, you do not expect those fields. So there has to be something else. So what can be the biologically relevant field that can act on the nucleosome? And the claim I want to make here is that this is the intranuclear pH, plays the role of the field. So now, qualitatively, why would you expect the nucleosome stability to depend on the pH? Well, qualitatively, let's say you have protons floating around in solution, you have your nucleosome. Proton binds to the core, obviously changes the charge, and so the DNA, core to DNA attraction increases. And this is very qualitatively, this is the origin of the pH effect. So basically, the qualitatively lower pH is supposed to result in a stronger core DNA association. This is qualitatively. Now, quantitatively, how does this work? Now, as we all know, there are a number of amino acids that can change their charge state depending on pH. Here are the key players here. Now, the normally at pH 7, this is their charge state. But if you vary the pH, if you bring it up or down, the charge state will change. So these are the players. This is why the core of the nucleosome responds to, the charge responds to pH change. All right, but this is just the core, so this is the charge. Now, how does it work when the DNA binds to the nucleosome? Now, as you will see in a moment, when charge state, when the state of protonation of any of these residues change, that can lead to dependence on the binding affinity. Now, why would the charge state change when something binds to the nucleosome core, like a DNA? Well, here's a scenario. For example, if you have a glutamic acid happy in solution, you block it off with a ligand, and then the charge state is no longer happy seeing water around it, that it sees a low dielectric medium, its charge state is going to change. Of course, the story, this is not the only mechanism, but this is how the pKs of titratable residues can change in response to ligand binding. Now, fortunately, this is one of the few instances when an exact relationship exists in biophysics. This is what's called so-called linkage relationship. Now, if you know exactly how the charge of your protein, ligand, and the complex, how they change upon ligand binding, you can calculate exactly how the protein ligand affinity, the binding affinity, depends on pH. This is this linkage formula. So what we need to do is to just estimate the charge of the nucleosome in complex with the DNA, and then the core and the DNA separately. Obviously, this is fixed. Compute that, and from this, you can estimate the dependence, the pH dependence of the nucleosome stability. So, if you examine this formula qualitatively, the conclusions you can make that in order to see a dependence of the binding reaction, to see dependence on pH, you'll have to see a significant changes in the protonation in the protonation state changes upon ligand binding, or significant changes in pKs in the range of interest. So either of the two must happen in order for you to see the dependence on pH of the binding affinity. Now, these things are hard to measure. Fortunately, these days they're fairly easy to compute. There's a number of ways to do it. We use our own server that computes pKs of proteins in combination with a ligand, with or without ligand. Again, I'm glossing over the methodology here. So back to the results. Before I go into the nucleosome, I want to show you some of the results on protein-ligand complexes in general. And the reason I want to do it is because there's really no data on the nucleosome. There's no quantitative data on the nucleosome. I want to validate the predictions on where the data exists and then take it to the nucleosome. So protein, DNA, protein RNA complexes, we've selected 20 of them, we have high resolution crystal structures of both the apo and the holo complexes. As you see, most of them belong to the nucleus, so they're relevant. So we'll look at them and look at whether pKs change upon DNA binding. And indeed, they all do change. They change by quite a bit, and they change in the one direction that, in fact, predicts of what is going to happen. Here is a typical protein DNA binding affinity curve as a function of pH. This is your delta G, this is your pH. And so you see that invariably, they all look pretty much the same. This is just one of them for a transcription factor. What you can see is that as pH grows, the complex becomes less and less stable, and at some point it becomes unstable. Now, where this point is, of course, depends on details of what the complex is, but this is a very generic curve, and I'll comment on these things later. So the general prediction is that increase in pH destabilizes pretty much all protein DNA complexes. Now, how does this compare with experiment? Again, there are few experimental points, but at least there are some, and some of them are, at least one of them is quantitative, so we can make a comparison. Now, for lambda repressor, there's quantitative data. So what we're looking at is a very small region of pH, and again, you'll see in a moment why. We're computing the change in the protein DNA binding affinity in the response to pH change of 0.3. Experiment predicts 0.777 KT. We predict 1.1. It's not too bad. Certainly enough to go on, certainly qualitatively and semi-quantitatively. Now, there is a number of other experimental points. Most of them, as I said, are qualitative, but all point to the same thing. So increase in pH destabilizes protein DNA complexes, and that confirms qualitatively what we see quantitatively in the calculations. Now, back to the nucleosome. What we want to do now is we want to just calculate the delta G of the DNA core association, but now we want to look at pH dependence of it using the same methodology. Again, you compute pK changes. They are large. They're all in the same direction. And it points to, again, the same thing. So you expect you see the positive pK shifts, and you expect the same thing. You expect the nucleosome to be destabilized as the pH increases. So here's the curve. Not surprisingly, nucleosome, again, it's a very generic curve for any protein DNA complex. This is for the nucleosome. Now, the slope is pretty steep. It's actually 25 kcal per mole per pH unit. So this is what we get. Again, no surprises there. The key question is, okay, so if you stop here, you actually pretty much made no connection to biology. The question is, does it all matter? May not matter at all, because we know that pH is really very highly controlled inside the cell, right? The cell cannot allow all, the biochemistry 101, all the buffer systems that try their best to control the pH, to maintain the pH. Does it matter for in vivo cells? This is the key question. Now, I would argue for the rest of my talk that it does matter, and actually does matter a lot. Here's a couple of points of evidence. If you look at the cell cycle, it turns out that along the cell cycle, pH varies by about 0.4. So that, again, this is not my work, obviously, but there is a variation of intranuclear pH along the pH cycle. Now, what about other processes that are naturally or unnaturally occurring in the cell? Here are a couple more examples. Now, in cancer, in this particular, I think this example was for breast cancer, pH, intranuclear pH increases by 0.3. But what's remarkable in apoptosis, there was a paper in the 80s in PNAS, in apoptosis, pH drops by about 1 unit. That's actually a lot. In fact, in that work, they concluded that it is the pH drop that drives apoptosis. They didn't specify the mechanism. They didn't know what the mechanism is, but through very careful, clever experimentation, they showed this is actually pH change. pH drop was a causal effect of apoptosis in cells. Now, so what I want to do now is I want to define a conservative value of pH change which is relevant to in vivo, and I call it 0.3. I say that 0.3 is something that you can see in vivo in the cell nucleus. In fact, it's not that tiny. It actually corresponds to doubling of proton concentration. It's not, 0.3 seems like a little, but units, right? So proton concentration is a factor of 2. So what we can do now is setting saying that, well, that's why we want to compute delta G of 0.3 and just see what happens, right? We can compute it through the linkage formula. This is just this integral. Simplified, essentially Taylor series, right? So what we want to do is estimate the nucleosome, estimate delta G of 0.3 at intranuclear pH. So, and just look at it. So we'll do that. So we will do that, but before we actually go on, so we will compute it, but we need a threshold of biological significance. Okay, we're going to get this number. But is this going to be significant? This is the second claim I'm going to make, is that I would argue, so we need to compare it, something to compare it with. I would argue that, of course, staying in Boltzmann Hotel, you would think KT would be the unit, but I would argue that's actually half KT is relevant. And the reason being, the half KT change and the transcription factor residence time can lead to a 3-fold change in the transcription rate. And for the nucleosome, they've actually also shown that about that 1.5-fold in the DNA accessibility can lead to, I think, the doubling of the transcription rate on the nucleosome. So I would say that 0.5 KT is the relevant, you would see, biology would feel it, 0.5 KT. All right, for the complexes that we did before, 70% of them, the delta G at 0.3 is higher than half KT. So I would say that for 70% of them, that dependence on pH is biologically relevant. Back to the nucleosome. This is the calculations for a few organisms, and these are the key methodological details here. Essentially, what you get is 10 KT as the lower bound. That's quite a bit, certainly compared to half KT. Now, if you look at details, strain of the ends of the nucleosome, again, delta G 0.3 is 2.2 KT. Again, this is also not insignificant compared to half KT. And so the predicted biological consequences So of course, this is all speculative. This is all theory, but yes, we speculate that whatever the processes that depend on accessibility to DNA is going to be up or down regulated due to the variations, naturally occurring variations of pH. So the accessibility will correlate positively with pH variation in the cell cycle. Now in cancer cells, that the pH increase is predicted to lead to more accessible DNA and possibly more active DNA replication. And conversely, we propose an explanation for why pH drop in apoptosis actually causes apoptosis. Well, the nucleosome just freezes. One drop on pH of 1 unit is a lot. That's the explanation. Now back to the hydrogen atom, putting it all together. It's not just my work, but work of many others. If you were to come up with this diagram, we just contributed to some of this. And the last bit I talked about is that you have your Stark effect, which is actually pretty stark. Nucleosome, the splitting of the energy levels due to pH. Thank you.